Bonjour. Merci beaucoup. Vous êtes ici avec la neige. So we'll be in English, but maybe a few questions at the end I can take in French. My name is Thomas Craig. And that's the title of my thesis, The Effect of Mycorrhizal Fungi Associated with Willows Growing on Marginal Agricultural Land. But before I get into what are mycorrhizal fungi, what kind of willows we're talking about, and what we mean by marginal agricultural land, I just wanted to give a few slides to share how I began this academic journey and give us some context. So I actually started as a geologist at undergrad, and this is a photo of my professor Tola Zanstad down in a South African gold mine. And my third year at university, I started working with him and spent a lot of time in these mines. And that's streaming hot water coming out. We were actually sampling extreme bacteria in these mines. And so I became fascinated by microbial ecology and especially using some of the new DNA fingerprinting tools that allowed us to study these organisms really for the first time. And later, as I was thinking about graduate school, I was interested in maybe looking at microbial ecology, but in systems that are more applied to our everyday life. And when I came to Montreal, I met Michel Adret and Marc Saint-Arnaud. This is a photo of Michel at a field site with these shrub willows, and Marc with a couple photos of mycorrhizal fungi. So we need to talk about what those are real quick. Shrub willows are a tree species, Salix, but they grow without a big trunk. They have all these multiple tall stems. And what you can do is called coppicing, and you can come in after it grows for a few years, you can cut it down, harvest that woody material, use it as a greener, low carbon fuel energy source, and it will actually regrow in a few years. You don't have to replant it, it regrows from the stumps. And you can do this for decades before you have to replant. So it's, but you're using agricultural land. There's concern for using food crop land. So the idea is maybe to use land that was settled for farming a century ago or more, but isn't now as competitive or valuable for food crops. And another concern, they grow, really quickly, but they take in a lot of nitrogen fertilization. And if you're concerned, our greenhouse gases and use of fossil fuels, well, a lot of synthetic fertilizers use those. So that comes to our other area we need to know about. So mycorrhizal fungi are fungi that live in the root area, the rhizosphere of plants, and the white, it looks like roots, but it's finer thread-like fungal hyphae that associate with these plants, and they associate with almost all the plants you see out in the world. And they interact and they provide nutrients from the soil, nitrogen, phosphorus. They can also draw a lot of water. And in return, the plant gives them carbon, a sugar source. And this is kind of a classic photo from companies that want to sell you mycorrhizal products for your garden, but they really have been shown to you can have in a lot of conditions a lot more growth, and it's been shown time and again that they can benefit plants. So there are two types we're mainly concerned with in this project. I abbreviate as AM, the arbuscular mycorrhizal fungi, and EM, the ectomycorrhizal fungi. They're, for the purposes of today, they're fairly similar, just that the arbuscular actually interact with the root and go into the cells of the plant, which is a little bit more intimate relationship, whereas the ectomycorrhizal outside with some surrounding the cells but not actually entering them. But we were talking about shrub willow agriculture, agriculture in general, and some of its issues. And when I met Mark and Michelle, they had this fascinating project they were telling me about, supported by the Quebec Ministry of Agriculture. And they were partnered with this Allard family that has a farm up in Saint-Marc-de-la-Chidon. This is an aerial photo. As well as a private company, Biocimiterra, owned and operated by Peter Matuidi, who could provide us with enough inoculum, and then the farm provide us with the space to actually test on a farm scale with the number of plants in a true life, realistic experiment, whether we could improve shrub willow cultivation, the SRC, which is the short rotation coccusing, and improve it to alleviate some of those concerns we talked about. And so I took on this project with them. Here's kind of a synthesis of how I viewed the project, because it really, mycorrhizal fungi can work with a lot of crop species, and overall, this project, I see it as informing all of agriculture through the lens of soil microbial ecology, and in this case, focusing on mycorrhizal fungi. So we have some practical objectives that the experiment had, that we planned and decided we needed, and which was we needed to measure the effect of the mixed inoculum. So we were adding AM and EM species to these fields in a controlled manner, in a controlled experiment, to see if we could benefit the growth and yield of these willows. We wanted to see how this effect was influenced by soil type or fertilization, because we wanted to better use marginal land, hopefully with less fertilization. And during the experiment, we wanted to verify and monitor the inoculated mycorrhizal species, as well as any native rhizospheric fungi that could be present and in competition with our inoculum, using these molecular techniques, which is that kind of DNA fingerprinting, extraction and sequencing. So this presentation today, I'm going to quickly mention some of the literature, talk about the experimental design and the methods. My thesis was split roughly into two papers within the larger series of chapters, and the first paper involved a lot of the growth data of the trees, and the second paper was more heavy on the molecular results. So that's how I'm going to continue to organize it, and then discussion, conclusion, questions. So start with, willows have been shown to interact both with EM fungi and AM fungi. That was important, one of the reasons we chose. And more evidence with EM fungi, kind of lower instances with the AM, but it's also, we've seen that small amounts of AM colonization can have a big effect. So other people have also done experiments to, try to work with AM and EM fungi in trees. Some in the field, but what we found was most of these experiments, when you grow a certain number of plants, you really don't want to have a huge number die from virus or a random problem. So you usually use sterile potting soil for your control, and then your treatment is sterile with your inoculum added. And so our study was really, from what we, surveying the literature, especially when we started, was the first that really had this very realistic conditions where the willows were put into the soil The control were with all the wild fungi, and the treatment with the wild plus our inoculated organisms. It's true, over the course of this study, I did come across a few other times. Mohammed Hijri has a paper where they were looking at farms that were using mycorrhizal inoculation, and that would be a little bit more realistic, obviously, but it was more a survey of actual farmers that couldn't quite divide up the land as much as we could for a formal experiment. And so we really feel that this work was pretty unique and valuable. When we designed it, our thinking, our predictions were that, we predicted the inoculation would positively affect survival and growth of the willows. We believe that the effect would be stronger on the more marginal fields, the less rich soil. We believe the effect could also be accentuated by the unfertilized treatments, but this could be complicated. It makes sense that the mycorrhiza should make a bigger difference where the willows are stressed, because their job, they are helping bring nutrients. So, but if there's a bit of phosphorus that they're limited by, but then you're adding nitrogen, and suddenly the mycorrhizal fungi can use the plant can use both. This effect we knew could be a little bit more complicated, but we didn't expect a difference between. We were going to use 2 willow clone types, but we did want to include them so that if there was a random insect attack or disease, we wouldn't lose all of our plants. Finally, we didn't, these willows, they're cut from the stems and they grow from the stems, so they're all genetic twins of their clones in a way of their parent tree. So, we are planting these fields with the same, two willow types, identical across all the fields. So there's a lot of theory that the mycorrhizal fungi associate with certain trees, and in this case, we predict that if they're the same willows, they should have very similar fungi. So we had 3 fields that the farmer helped us identify as his marginal land types. We had 2 willow clones of the species Salix miabina, the lineage SX6164, which Michelle's group had identified as growing very well in Quebec. We had 2 mycorrhizal strains, the AM, the Rhizobomus irregular, very popular in all the kind of products you buy at the garden stores, like Mike, peat moss, and things like that, as well as this EM strain, Pibuloma longicatum, that Biosanitera was providing us with. And then we're working in farm fields with all of their variation, and so we had to divide up our land in a very rigorous way, block it out, apply treatments within treatments in a very rigorous way. So again, this is the map of the field. We had the 3 fields there. This is the way we kind of were working with the maps when we went out there, and so it's flipped north to south because this is where we were always approaching the fields from the farmers' parking lot and barns. And this plot we came soon to start colloquial calling the rocky field, because the farmer chose it. It's marginal because it's difficult to get equipment through it. It has a lot of stones on the surface. This here is a very deep, kind of a few meters wide by a couple meters deep canal almost to take all the drainage from these fields, and which is helpful for growing crops. But especially the years we were conducting this experiment, in particular the first year, we had issues with extreme drainage and drought conditions almost, especially for our willows. And so we started calling this our dry field because it was suffering from that problem. And back here was almost 100% sand, so we were calling this our sandy field. And it is extremely bad for farming crops. I mean, waterlogged next to the forest, very low pH of 6. You know, it was had a very marginal history with the farmers and the farmers' experience. So, like I said, this is just showing 4 of the blocks out of a plan of 12 blocks for one of the fields, because each of the 3 fields was randomly divided differently, but they all had these 12 blocks where it would be split in half, with the blue in plus had the inoculum applied, and within that, half was fertilized, F plus, half unfertilized. It was, this is the hierarchical design, but it was also structured in that the first 3 rows would be one type of willow, and then one of our lineages, and then the next 3 rows would be our other lineage, because we had to work with the limitations. We were putting out tens of thousands of willows, and so we had to use the farm equipment, and it's hard enough to start and stop the inoculum delivery, but to also change out the willows, we had to make sure we had the same ones down the rows. This is the picture of the planting. We actually applied inoculum. We had a water reservoir in front, and and the inoculum would be let out at each point where the plants were, the little cuttings were dropped into the soil, and that could be turned on and off as we walked down the rows and reached, we've got the map. It's hard to see, but there are flags at each of the 18 meters that we're starting and stopping treatments. These are the cuttings. The farmers are dropping down into these cups, and then they go down into the open soil and then close back. It's a little harder to see. You can see the cutting in the soil there. This one was actually picked up a little bit to see better in this photo. Most of them are pushed down. They can be pretty under the soil and still sprout. So this is a little small, but I was trying to fit it all in. It helps us just look at the whole project. It was over 3 growing seasons. We had to sample the soil, we had to plant. Then after a season of growing, measured the height and diameter, as well as digging up some of the roots for our DNA analyses. And then that first fall, the standard practice is to cut it, to coppice it, and then we applied the nitrogen treatment that second spring, which is again kind of standard the way these are farmed. After the next season, we measured height and diameter as well as weighing biomass, which was significant. You have to get in and cut them, weigh individual plants, record that, and dry a portion to make sure you know how much moisture content's in the willows. That second season, it was not coppiced, so the measurements the third year are cumulative over 2 years of growth. There's a few more details. Every 36 centimeters, these cuttings. The inoculant was applied at roughly 350 propagules to 250 propagules per plant in 50 mils of water. The nitrogen source was pretty much pure nitrogen from composted and pelleted chicken feathers. At a pretty standard rate for willow growing. And another detail, that first year we did have pretty intense drought conditions right after planting, and almost 25% died in the dry field, 15% sandy and 5% rocky. Luckily they were scattered enough random that I think only one block was compromised and we were able to sample accordingly. But we did have to take that into account when calculating the yields, because you would have to account for the lower density, and so a lot of the yields are given per hectare, and that takes that into account, because those plants left in the dry field obviously have more resources because some of their neighbors are dead, so they can grow a little bit more. This was that first fall. You know, the trees are growing roughly waist height. We had to dig up. We were digging up entire root rhizospheres, like big root balls. This is the second year of growth. So this is just, you know, it had been cut down. This is 1 year of growth. You know, it had had the roots left from the year before, so it had a jump start. But it is impressive, this 1 year's growth. And you can already see, like this year and probably this year are one of the fertilized treatments. You can kind of see how it's a bit, jumps up a bit. They just take the fertilizer you give them and really grow. So overall kind of summary to get an idea of what we accomplished. This was at farm scale. It was with farm equipment. We had 21,600 trees that had not been fertilized. We measured the first year, 2,240 trees. It's a lot of effort to dig. A few people that helped me dig for roots are in the audience today, and so the first year we dug fewer, but then that next year we actually dug up 96 rhizosphere samples, so root samples, which was a very intense effort. And then we at the next year that we cut and weighed quite a few. We took measurements for fewer because it took a lot of time to cut and weigh them, and so we only had time left to measure fewer. But these were random samplings from within the larger experiment that had the tens of thousands of trees. So now we can go to some of the growth data, and not to leave you in suspense any longer. We did not. This was a big surprise for our experiment. We did not see a benefit from the inoculation. I mean, this is about as this last year we didn't have the number sampled was smaller, and this is pretty much flat within the amount of variation we see in these fields. There's a lot of the standard deviation is high in this willow data. So I mean, between uninoculated, inoculated, and these letters are showing the results from the statistical test, which verified, we found no result from our inoculation. And this is based on the diameter measurements. You calculate the area in meters squared of the stems. It's pretty standard in forest research. You also kind of to estimate the amount of growth and biomass. But this is roughly. I also have just to give you an idea for people that work with biomass. This is equivalent to the oven-dried tons, just to give you an idea of how much this stem basal area per hectare is. But this is just an example. We ran these statistical tests on each year on both the stem data, the height data, and the weight. So we had to run these ANOVA models, these ANOVA statistics, analysis of variance, on each of those cases. And it was a pretty complicated model because we had to look for a field effect, inoculation effect, fertilizer effect, and an effect of the line of willow we used, since we had used the two. And then all the combinations of those four things, because we also wanted to see if there was an interaction between, maybe inoculation would work in one field but not the other, so we had to test for that. And this just is to show, similar to what you saw in the graph before, there was no inoculation effect. We did, though, see, a difference, a strong difference, a statistically valid difference in the fields, and it's expected that fertilization would show a strong effect too, but it's good that that was verified. Just to see the effect in the fields, it was pretty interesting because the sandy field, which had been so marginal for crop growing, food crop growing, was actually right off the bat, amazing for the willow growth. I think they really grew well, responded well to the moisture, the heavy, almost waterlogged nature of that field during parts of the year. They could handle that low pH, and so the sandy field really grew had several times greater growth the first year. The other fields started catching up, and by the third year it caught up. And a lot of this also seemed to be from, you could just see when we sampled the roots that in the sandy field, the roots could spread out really quickly and could access all the space and nutrients. And so they, in the clay-rich, medium loam soils of these two, they took longer for the roots to penetrate and develop, but they eventually had access to a similar level of nutrients. And also just to get an idea of how much of a boost the fertilization gave. Okay, so real quick, and this in parentheses because it's not part of the larger field experiment, but it's important that I convince you that, okay, well, the inoculum didn't work, did we apply a living inoculum? And we did take portions of our inoculum to the greenhouse. We have a few willows that we inoculated in sterile soils, like we taken farm soil and sterilized it. And so, we were doing a bit of microscope work, not enough to test, rigorously colonization levels, but just enough to see, there were AM fungi, there was EM fungi in the from the viable inoculum. Also, one of the other grad students working in the greenhouse saw, presumably, and he did some taxonomy. Mushrooms come up from the EM inoculated plants, and they did. It was a little inconclusive because they were definitely a Hebeloma species. They couldn't identify them to longicaudum, but this was again a good evidence for the fact that our inoculum was viable. So, really quickly, I do want to show you a little bit. Again, it's not from it's not the main point that we're looking at inoculation, but I do want to show you the amount of variation we had in these fields. So this is the dry mass from just the rocky field, and versus the cross section. And this, I made this graph mainly because I was making sure it's a lot faster to measure diameter and then cutting a plant and measuring the weight. And so if you can, and they do correlate well, have a R-squared fairly high, this means that you can go out and measure the diameters, and your stem basal statistics are pretty valid for what you want, which is the yield, which is the weight, the biomass. And but I'm showing you this graph mainly because I want you to see that we had a lot of variation in these fields, which astounded me because, again, we're working with willow cuttings, essentially clones of the parent plant. So the same exact genetics, the same field here, and within the variation of, maybe a little bit more water in one patch of land, a little bit more historical nitrogen or phosphorus or what have you, a little different texture. One of these plants could be 50 times bigger than another plant, which was very, which also spoke to the difficulty in a field experiment like this to see a small effect if it was there, because you're dealing with such large variation. So now to the molecular methods. So I have a few of these in parentheses because we did a lot of work that did not end up in the thesis, and even and certainly couldn't be included today because I wanted to give you the basic picture. But we had it's a known difficulty that when you're sampling with these DNA fingerprinting techniques for mycorrhizal fungi in the rhizosphere, there's a lot of inhibitors in the soil, in the root material. You've got a lot of root DNA getting in the way. It's still an ongoing problem, and we spent a lot of time troubleshooting. We also, I would have loved to have a master's student working with me, or I had the help of several students over the years, but we still didn't have time to do a full survey of colonization rates, but we did quite a bit of staining and microscope work just to, with the idea that if we had time, we were going in that direction. But in the end, we spent a lot of time on our molecular techniques, and we did a full run of DNA extraction, cloning sequencing, looking at the 18S gene and ITS gene, making these likelihood trees, searching the sequences in the database, and a lot of that did not. I'm not going to show you today just because it was less convincing or less conclusive. In the end, the very last year, we had the help of a couple other. We did have a postdoc and a couple other people at the facility that were doing more high throughput sequencing and on one gene, and we did get a much stronger data set. And so that's what even though a lot of the years of work was a little bit different, I really focused in my thesis on these results, and they were, so let's go to some of those. We did not find the inoculated species, and this kind of helps explain some of our field growth data, and that we found a lot of wild fungi in these samples, and it really looks like and a lot of unusual fungi. These high throughput sequencing techniques can have millions of sequences. They're very powerful, but a huge chunk of the 264 of the 702 fungal OTUs, we could only say they were fungi. We don't even know if they were Glomeromycota, Ascomycota, Basidiomycota. It was quite astounding. The most dominant fungal sequences were unclassifiable at the genus and species level, and many still were at the family, order, and class level. So I'll talk a little bit more about this, but just to show you, this is an analysis that plots plots your samples based on what the diversity, what species are in them, and how many of each. And it basically you can differentiate them by inoculated, uninoculated, and there was really no difference in these wild communities. We didn't really see, but what we did find, which was pretty fascinating, because as I said, we were using the same willow, the two types in our three fields, and the sandy field had a very distinct community of microorganisms, of fungi, compared to the rocky, and in this case it was supposed to be dry, but that field, we kind of changed its name over the years because it it was a puzzle to us. But this shows you, again, that difference between the sandy field with the low pH. That's another thing. Between rocky and dry, dry field had a pH of 7, rocky had a pH of 8. So these fields were pretty different. Also, dry had twice the total phosphorus levels of rocky. So it is interesting that these two fields were very similar, and in fact had the majority of the species were Ascomycota, whereas it flips in the sandy field, and the majority are Basidiomycota. And we have to correlate that. We can only point to the fact that what we saw, the main difference was soil texture, which associates with often moisture availability, which is just a huge driver for diversity. This is the same, roughly the same graph, but taken down to a little bit finer level of, the same Basidiomycota and but one of the more common species, this Pyronematocea, it's closely related to the Physalis, which is a known mycorrhizal fungi. Also, this Cortinariacea is closely related to this Inosi, which is a known mycorrhizal fungi. So, we were sampling the roots of healthy plants. It makes sense that we were finding a lot of mycorrhizal fungi. And this, I did want to show you, it's not laid out immediately clear, but it was just such a fascinating, from the first year, I was showing you the second year's molecular data. This is from the first years where we actually did cloning sequencing, which took a lot more time. And could do fewer samples, but gave a bit longer sequence reads, and was still pretty interesting in that. The three fields, so this is the dry field, the rocky field, and the sandy. We didn't find any AM fungi in the sandy soil, so we had to look in the sandy roots. That's the difference between these two. This is using a nested PCR where you kind of amplify for it, and you take that result and amplify again. So it's really looking at minute traces, and we couldn't do that with the high throughput data yet, or we had to concentrate on our ITS. But in that first year we did find AM fungi in the first year, and it was fascinating that across these three different phyla, like Glomeromycota, order, Diversospora and Ambospora. These are very, very divergent groups, taxonomically divergent groups of AM fungi. And it was just amazing that they actually seemed to partition out based on the three field types, that the soil could make that big of a difference. And I have this up just in case we have questions later, but I talked a little bit about the difference between the dry field, rocky field, sandy field. There's a lot of information, but pH, the soil texture, total phosphorus. I think I hit most of the high points, so if we want to, we can go back to that. So the results we summarized. So inoculation did not positively affect survival and growth of the willows. We did check survival too, and it was too bad that the severe drought conditions happened the very first month before the mycorrhiza had a chance to establish, because maybe we could have seen a survival difference, but it was right in the beginning. The cuttings either sprouted or they didn't. So, but we didn't see the growth that we expected. Soil texture had a profound effect actually, and we hadn't really predicted that. Not plant species, nitrogen fertilization, soil pH, or these other. Also, it seemed like that second year, the ITS data, we didn't find any AM fungi, so it really seemed like the AM fungi appeared to dominate, not the AM. We can talk more about, it's a little inconclusive, but I basically look at the results, and I would say that our current inoculum species selection inoculation techniques appear flawed, at least for inoculating in wild, intact ecosystems. Probably if you have completely, I mean, it's been known to help in sterile conditions like mine tailings, or you have a new development and you scrape off all the topsoil, or you're making a raised bed garden and you're using just sterile soil. But in the fields seem to have enough wild organisms that the ones that we're using, that are available to us today, do not seem to be competitive. And the fact that our molecular results found all these unknown fungi, really ones that we're not growing in in labs, really point to the fact that there's a disconnect. There's a lot more to be accomplished. And finally, the soil texture appears much more important than previously appreciated. So, I really feel like it's up to us to do this hard work of developing new culturing techniques. Possibly, working with, instead of just, we've been confined to potted soil that, doesn't have a lot of disease and has a uniformity, but since it's so important, we might have to work more in more field realistic soils to make inocula. That's a challenge, but also maybe semi-sterile or unsterile, which wasn't possible in the past because you couldn't say for sure what was there, but with modern sequencing, we can probably assess as we go. And, this is another thing we really need is, the molecular tools are so powerful, but they really have limitations. Like, they can kind of tell you what's there, but it's hard to know in what numbers, to what is left over in the field from past years of plants, what's active. So to get these kind of things that are important for classic ecology, to really kind of work with the modern molecular techniques and to develop that. I mean, these are all hard things. I mean, we're all working on it, but I just want to point out what I really came to appreciate through my study. AM seems to have a difficulty in doing the DNA extraction or the PCR. There seems to be a bias. I've seen papers even recently where looking at other genes, they suddenly started finding AM fungi that had been missing from previous surveys. So there definitely is some room for finding these. This is, I guess, in modern agriculture, we do monocrops. We go in and we weed, and we have the species we plant, and we can work with them over thousands and hundreds of years now and now it's even accelerated to breed our perfect crop plant. And so it only makes sense that there's going to be a temptation to possibly sterilize and apply soil microorganisms that we've bred, but that's an extreme change. So we have to really think more carefully about what the consequences of that are. Find out more about what is there wild, how we can work with it, maybe without quite weeding and planting in the traditional agricultural model, and see how we can kind of nudge it towards what we need. But those were some of the things I really came to appreciate. So I'd like to acknowledge my advisors, and we had lots of people helping me over the years. Some of them are in the audience. I appreciate you guys being here today. And And I'm open for questions.